Hi everyone, my name is Pineo-Cavanaugh, and today I'll be talking to you about genetic and hormonal factors in Alzheimer's disease risk. So first, I just want to briefly summarize the lecture. We learned that there are two forms of Alzheimer's in humans. The first is the sporadic form, and that's over 99% of cases and usually starts after 60. Then there's the familial form. This is less than 1% of cases and has an onset around 30 to 60. And the familial form can be caused by mutations in the genes APP, presenilin 1, or presenilin 2, all of which lead to excess amyloid production. So you might then think that amyloid is at the root of Alzheimer's symptoms. However, research findings have complicated this theory. Firstly, we know that amyloid levels peak early on, but remain stable as symptoms worsen. Secondly, amyloid imaging does not regionally predict where the brain is sick. And then lastly, trials that have used antibodies to clear amyloid plaques have not seen resultant symptomatic improvements. So it seems like the plaques are not the root of the damage. Maybe it could be the oligomers. And then, interestingly, tau PET imaging does regionally predict damage in Alzheimer's brains. That being said, typical transgenic mouse models have both amyloid and tau aggregates without showing progressive neuronal loss. So, what can we learn about the cause of Alzheimer's from sporadic patients? Well, genome-wide association studies have basically identified only one clinically relevant gene, and that's ApoE. The ApoE E4 isoform increases your Alzheimer's risk from 20 to 65%. It shifts the onset earlier, and it's associated with greater amyloid deposition from an earlier age. However, this effect is attenuated in African Americans, and it's also modified by sex. So in women, the E4 isoform basically doubles your Alzheimer's risk, whereas in males, it barely changes. And then besides ApoE, we have found a few other variants with much smaller effect sizes. For example, there's TREM2, and this has an intermediate effect, maybe because it's related to neuroinflammation. And then there's also this heritability problem. So collectively, the SNPs identified in GWAS can explain 30.6% of the variants. However, we know that 53.2% of phenotypic variance is genetic. So why is there this discrepancy? Well, Dr. Grecius pointed out that many of these studies lack gene-gene interaction analyses. Additionally, the SNP arrays only really catch small mutations. They don't catch these large structural variants. They can also often be underpowered or not have diverse subjects. And then finally, a lot of them don't include mitochondrial DNA or omit the X chromosome, which is surprising given the sex differences that we know about. That pretty much concludes the lecture summary, and now I want to briefly talk about why I'm interested in Alzheimer's. I learned extensively about this disease in Dr. Sapolsky's seminar in the fall, and I found this content really fascinating, not only because of my academic interest, but also because I had been helping take care of my grandmother, who had late-stage Alzheimer's at the time. So I was really surprised to learn that estrogen is a protective factor against Alzheimer's, because my grandmother's Alzheimer's had started shortly after she started taking estrogen suppressants. So I wanted to use the remainder of the presentation to explore estrogen in relation to these Alzheimer's-linked genetic variants. The first study I want to talk about is by Wang et al. So this group looked at the effects of activating estrogen receptors alpha and beta in rat hippocampi on ApoE expression, and they found that estrogen receptor alpha activation increased ApoE expression, but estrogen receptor beta activation decreased ApoE expression. So given ApoE's relationship with Alzheimer's, this finding made estrogen receptors kind of an interesting topic in the Alzheimer's field. Then the next study I found was by Bove et al. A few years later. This group recruited 1,884 women, and 32% of them had received surgical menopause. And so they saw that in the surgical menopause group, an earlier age at menopause predicted a faster decline in global cognition and increased AD neuropathology, particularly in the plaques. And they also found that hormone replacement therapy within a certain time period attenuated this cognitive decline. So these findings kind of pointed to some interesting estrogen-ApoE interactions that helped fuel further work within the Alzheimer's field. So then, more recently, Ratnakumar et al. Assessed genetic expression changes in overactimized rhesus macaques that were treated with estrogen or vehicle. So this is a model organism that's much more similar to humans. And they found that in the macaques, 504 genes were upregulated by estrogen administration, and 84 of those genes were also known to be downregulated in postmortem Alzheimer's brains. Moreover, the gene with the third highest estrogen-induced increase was none other than ApoE. Then the researchers independently analyzed exome sequencing data from a bunch of female Alzheimer's cases and controls, and they found 50 significant variants. And one of the variants they found in the human Alzheimer's cases was MCM8, which is notable because mutations in this gene can cause early menopause. Hence, this study kind of validates the menopause-estrogen link and implies that estrogen mediates this effect by regulating ApoE and other protective genes. After more research was starting to emerge suggesting that there could be this link between estrogen and ApoE, people started to look at genotypic interactions. Kunzler et al. Did a study where they gave estradiol to EFAD mice, which express human ApoE and accumulate high levels of amyloid. And they saw that estradiol administration decreased both insoluble and soluble amyloid beta in the ApoE2 and the ApoE3 mice. However, in the ApoE4 mice, it decreased soluble amyloid beta, but increased insoluble amyloid beta. So this is helpful information for if we want to kind of think about estrogen as a therapy for humans in the future. And this leads me to my takeaways. So to recap, we saw that menopausal loss of estrogen is associated with an increase in Alzheimer's risk. And then we saw that, at least in the mouse models, estrogen decreases all the amyloid in the ApoE2 and ApoE3 mice, and it could be a good therapeutic target, hence for humans who are ApoE2 or ApoE3. However, because estrogen increased the insoluble amyloid in the ApoE4 mice, we probably need more research on soluble versus insoluble amyloid toxicity to understand whether this could be a good therapy for human ApoE4 carriers. So here are my citations, and thank you for watching my presentation.